Okay, good morning. Let's get started. So this is week 10. We are going to discuss components and the decomposition. So the first part of the course, we will spend some time to reinforce your understanding of components in terms of the lineage algebra, and also put that into your content of your biology research. Then we will go through a series of decomposition methods. How can you factorize matrix into different components and see what they mean. So if you today, if you are giving a matrix, something like this, for example, in the Excel spreadsheet, you may collect your data either from a single-cell RNA-seq data analysis or something else. You always have a numeric table with rows and columns. I hope you can do more than before after you take this class. So what you can do right now with this matrix, you can, for example, compute the average of the columns, okay, compute the average of the rows. If you know what that means, depends on sample variables, whatever, you know you can do this. What else you can do? You can do transpose, okay? Row become columns. Anything else? What we have learned to do covariance matrix. So you probably can see, okay, I want to compare this against that. If you have multiple rows, you will compare pairwise distance. You build a matrix. You can do by variables to understand the correlation between variables, their values distribution among samples. So that's something you can do, right? But if you know this is example we did for the matrix multiplication example, you will realize this can be decomposed or factorized into multiplications of two matrices. So that's another level of understanding of what makes this matrix looks like this, okay? So that's what we are going to discuss today, to do the factorization. So what it is, if you have a number, say 21, so you do the factorization, and you know it's multiplication between 3 times 7, okay? So that's your decompose or factorize number into a product of the prime numbers. In this case, we are going to do a decomposition of this matrix. By chance, this is just a 2 by 7 matrix, but you can generalize this into n by m dimension, right? So we know we can decompose this into two matrices, which is A and B, okay? A times B. There are many, many different ways to do this decomposition. To make this make sense, we know the example we had was you and your roommate want to make a sandwich, and 3 types of them, and each sandwich require bread, lettuce, and whatever different ingredients and in there. So you have to buy how many pieces of bread, how many ounces of peanut butters, jelly, something like this. Depends on number of your sandwich you want to make, and also what you want to put into the sandwich, right? So, so these two matrices contains those information. So the matrix factorization is to, when you observed this end product of the matrix, so you only have the information that you want to buy this number, this number, this number. Your roommate want to buy this number, this number, this number. But if you want to go to the inverse direction, you want to figure out, you and your roommate, what you are going to make and what's the gradients in each of this sandwich you want to make. Okay, so this is opposite direction. As we know, to do the multiplication between two numbers to get a product is so easy, but when you do the inverse operation, you want to factorize this number. 21 is easy, but if the longer number take longer, and you know the crypto, this security numbers are encoded by the primary number factorization. If you have a longer number, it's hard to decode. So it's also much harder to go opposite this way. There are many, many different ways, so that's what we are going to discuss. Okay? So we want to have this concept re-emphasized, okay? Make the strong impact on your mind, then you can, when you see a matrix like this, not only take a mean or get an average number, rows, column, compare pairwise distance, you can also do the decomposition to factorize it in order to understand different components in response for make this and the product of matrix that you observed. Okay? Then we will go through NMF, which is called non-negative matrix factorization. And then we will revisit eigen decomposition. Okay? We have used this a couple of times in the PCA and other algorithms. And then we will discuss SVD, singular vector decomposition. And at the end, we will mention independent component analysis, ICA. So if you search the Wikipedia page, there are many different ways to do this decomposition. Okay, so decomposition is a factorization of matrix into the products of matrices. That's definition. There are many different ways, so we will mention a few of them, but they all have specific case, user case, that you need to do this, you need to do that. There are many, many of them. Again, the most important one, the SVD, is the most important ones. Okay, most versatile and with most robust algorithm people use in the engineering computing. This is just a revisit what I just briefly mentioned again in this first couple minutes. So you do the multiplication by put this in the position. So there's a 2 by 3, there's a 3 by 2, and because inner product dimension are matched, so you can do this multiplicate elements, then summation to get this position. And then you get this position until the end. So that's an example. So we assume we have 3 different types of sandwich. Okay. So let's use this example. So this end product can be considered as there are 3 layers. Okay. You can think about this will be, so this is a 2 by 6. You can think about there's another 2 by 6 matrix with numbers. And the superimpose onto it, there's another one. So I put in there's another one. So you have also, and then you have another one. So you can think about this like 3 layers of okay, 3 layers of this. So the summation of these 3 layers, if you look at the first one element, if you add them up, it should be 24. Okay? So the second, this position, and it's already overlapped. So this element add up should be 24. So this is another way to look at this matrix. All right? So the first matrix will be, what that means is in the here will be if you and your roommate only want to make type 1 sandwich, that's what you want to buy. Assume you don't change this number here. You want to make 4 and your roommate want to make 3. Okay? So the first layer will be that. And the second layer will be you want to make 5 and your roommate want to make 3. Type 2 sandwich, that's amount of stuff you want to buy. So this layer, what I'm talking about, is the so-called component. Okay? You have your element which different sandwich, that's your element, your components. And then for each component, you have an amount, which is your loading. You want to, how much your amplitude, or how much you want to make, okay, to put that component into the combination of the total. So the first layer is corresponding to the first component. That's your type 1 sandwich. Second layer is your second sandwich. Third layer, your third type sandwich. Okay? So this example is from Netflix. Okay? People have the company collected this huge matrix which users, okay? The Netflix subscribers, and I don't know how many. And then you have many movies, and then it make a sparse matrix, because sometimes user provide their favorite movie and put either the 5 star or 3 star, something like that. So you get the information about this user movie corresponding matrix. And this is sparse. There are missing values, of course. But the company want to decompose this into users with major components, see what type of users maybe, okay? So the D, you can define this latent variable based on what you need to do. And then you can classify the movies and using another matrix, which is D by number of movies. And the product of these two matrices, U and V, will approximate the values in the observed data matrix R. So this is one of the examples which different from our discussion of sandwiches. So we will first discuss this non-negative matrix factorization. So if you look at this visual illustration, you have an observed matrix, have a lot of samples, and then there are a lot of these variables, okay? In this case, it could be the samples of gene. The gene will be the columns, okay? So if you look at those samples, the first 1/3 of them, looks like there's a block here, right? So those samples express the first half of those set of genes relatively high, and then they shut down those genes. Well, this part of the samples here, okay, I don't know if you can see on the screen, looks like there's another block of samples which express this part of the gene higher, and then this looks like there are 3, we see components, okay? So you want to have that information simplified or write into a product like this. This column only have 3 columns, this matrix. So it's much simpler than the original observation. So this is only number of samples with 3 columns. 3 columns indicate those group of samples which express the first component strongly, and the second component and the third component weak. Okay, so this is indicated by this color difference. And this is the second group of samples. And in this matrix, which will make a product between these two to reconstruct original, you see it also have 3 components, okay, 3 rows. And 3 rows to give those genes different weight, okay? Those genes involve mostly in the first component. So you have high values in this right block. And then the rest those genes involved in the second components or gene program, what do we see? Okay, so these 2 simpler matrices, the product will reconstruct the whole observation again, okay? But we want to know these 2 simpler versions of these 2 matrices because we want to know the internal relationship between gene program, which components, and their relationship with the samples and the genes. Okay, we want to know what gene involved in which gene program and which samples contains or express more of this program of genes. So I mentioned NMF first because this is straightforward. We can understand this with the real samples, numbers. But in terms of the development of algorithm, NMF appeared much later than SVD, for example. SVD had been here 150 years, maybe longer. But NMF is only become popular from 1997, 1999, so it's much modern and current. So the reason is, this involved this optimization procedure, which with a constraint, it requires A and B contains non-negative values. Non-negative means you can have zeros, but not negative values. Anything above 0 is okay. So this is kind of constraint make the optimization procedure harder than free parameters without a range. And also, the application requirement is higher recently because of the gene expression analysis require gene expression is not always non-zero, non-negative. Okay, could be 0 but higher. And also the image processing, so machine learning field become realize that there are applications, many applications in non-negative matrix factorization. So that's why it's developed late and more there's more realistic applications in the real life. So the expression of this is, if you have observed matrix X, you want to approximate this X using X hat here, okay, to replace. It's very close, similar. There's some noise maybe in the original matrix. But you want to use two matrices, U and V, their products to approximate this original observation. So that's the expression of that. And with the constraint that all the elements in the matrix U and V has to be 0 or greater than 0. So that's the whole expression of this problem. So we can achieve this using an expression like this by looking at, so V is original matrix, and we want to get W and H to do the factorization. So we want to make the difference between these two as small as possible. So the distance we want to compute is the difference in, so if you have seen this is a norm between all the elements put together by square. So this is a distance based. So we want to make this function get the W and H value in order to get minimize the distance between their observation, the observed matrix with this factorized two matrices products as small as possible. We can also use KL divergence, okay, to compute the difference between V and HW. So whatever we want to make this, if you want to decompose it into W and H, you want to make them as similar as possible. Okay, how you measure the similarity between them, that's a cost function. So you can design your own cost function as long as it makes sense to make this look like that, okay, by changing the values of this. So in our case, we want to change the values in the matrix A and B to make the product same as this, okay? So the technique we used is to, so you see there H and W. So we first use the elements inside the W to express, to update the values in the H, and then use H value to update this. So this keep updating itself, and each steps you compare your end product with V and see whether the similarity get improved, okay? So they make distance as small as possible step by step. So the algorithm we use this to update by iterate the values inside W and H to make them at the end they match the observation. So this is a problem, and one is by distance, another is by divergence. They are the same, okay? Just a different expression. So this is update that I just described, to update the algorithm that is used to update the elements in the U, for example, okay, elements. So here we use a different notation, okay? So X, so UV. So the X So sometimes people use WH to approximate V, but here use UV to approximate H. So they are the same. So let me just rewrite this. I think we are okay with this, right? Okay, the different layers. So you don't have to fully, I mean, you understand this is enough. So that's inside the algorithm what they're doing. So you don't have to re-implement it because this has been coded up by many different languages. Okay, so we just want to know what's going on inside. So that's what you want to approximate U and V. So inside the U, this is a matrix with Uij inside. So this matrix. Then here you have Vij. Okay, so you want to fill up the values in these two. Okay, they have to be multiplicable, so you know the inner dimension are the same. But if you have I and j, you have a lot of these values. So you update Uik, okay, every elements in there using the information from V, from X, and then from the U itself. So in this way, you update the values, and then for the element inside here, you use information from here, and to update its values. So this is the step iteration, okay, so inside there. There are some theory which why this work and their algorithm design which involved in the start from you want to minimize this difference, right? You write this up into the trace of these two. So this is the same, okay? Then you expand this expression and get your likelihood function. So you want to minimize the whole thing here. Then their partial derivative. At the end, people derived the equation like this, okay? Based on those theory. So again, this notation change again. So it become A and WH. So still X and UV. You keep updating. After several rounds of updating, and those numbers won't change, so you get the values. So, for example, this matrix is decomposed, use non-negative matrix factorization, become this times this. Okay, so this red one is matrix W, and this purple is matrix H. I think is H transpose. Okay. So this is a this matrix is 3 by 2, and this is 2 by 5. So at the end you get a product 3 by 5. Any questions so far? So this is inside the source code. You will see if you decide to use Euclidean distance, so this is a cost function set up. So every iteration you get U minus U hat, which is V, and V is original observation. The V hat is your product of W and H. You want to approximate it. So you want to make this small. Similarly, if you want to KL divergence or other divergence as a distance, you can replace this function. Okay, so this is a place where the cost function is defined. In this side is the iteration to update the matrix W and V in order to let them to update matrix H and W in order to approximate this V. So you see those are the element-based multiplication. Okay, so because you have dot here, this is a MATLAB function. So you update every element. So here is matrix multiplication. So you see here there's a dot type multiplication, and here is a multiplication between matrices. But anyway, this just follow the equation we just showed here. Okay. When you do the element based, you have this dot operations in front. So you look at H was updated using information from W, H itself, and V. So keep updating, and also the W value will be updated using V, H, and some W itself from a previous step. So you keep updating. At the end, you will get converged results, and these two become stable, and you output the results. So this is MATLAB NMF function, the documentation. So it will take two inputs. So A matrix is what you want to do the factorization, and you need to give a number K. Okay. So the sandwich example, if you don't know how many sandwich you want to make, you may try two, maybe try three. The result will change. Okay. So if you try two, then your A-B matrix will be yourself and two sandwiches, and two sandwiches with their ingredients. If you're three, then there are three different types. So K is hyperparameters you have to know. Okay, in real application you may not have idea, so you may want to try different K. So inside this documentation, we see the norm of this matrix norm. So this is a difference between A and W and H product, and then normalized. So you want to minimize this distance. This is another implementation in Python in the package called the scikit. I don't know how to pronounce this, but it's a very famous machine learning package in Python. If you do this sklearner, then decomposition, their method, and MF. Inside here, the same. Okay. The implementation, same. This is one of example. Is real early applications of NMF in gene expression analysis. This is a paper published in PNAS, okay, National Science Society proceedings. So 2004 paper, they start to use NMF to decompose a gene expression matrix. At that time, there's no single cell technologies, so each columns here is a sample. It could be from one biological replicates, could be the different samples put together, but each one is a bulk RNA-seq, maybe not RNA-seq even, maybe microarray technique. But anyways, you get the expression of many genes. So you have n different genes with m samples. Then author assume there a super hyperparameter k. So how many components in this matrix? Okay, so they gave a name called meta-gene, or meta-gene maybe just gene expression program, what we just described. So meta-gene. Meta-gene in our term is the components, or maybe it's a gene expression program. Gene expression program is a set of genes maybe expressed in a coordinated way and to execute some function as a group. Okay, so you can think about that as a gene set which co-expressed involved in particular gene cellular process or signaling pathway. So in this case, we assume there's only 2 meta-genes from this n genes, and then the authors just say, okay, there are 2 meta-genes, and the red one indicate the genes by rows and their involvement in how much they involved in these 2 gene expression program. If you look at this gene, the old purple, then this gene evolved equally in 2 programs. Some genes maybe is blue in the first one, and red in another one that involve more in the second program, which other genes maybe in the opposite expression. So you get this W matrix tells you the involvement of each individual genes in the corresponding meta-gene expression program. Then in this side you have this gene expression program and the samples, okay, the components of samples, fraction of samples. Each sample get involved in. So some samples may have, you see this green indicate the second meta-gene, and those samples at the end will be classified as a number 2, and they have a higher involvement or expression profile in the meta-gene program 2. Will class 1 have a higher in this, and the class 2 have higher in the second. So this is an example of application. When we involved in this next generation sequencing and generate large scale omics data, matrix factorization, especially non-negative factorization, have been widely applicable in many different data mining tasks. Okay, so this is a review paper by Alina Fertig. So she's one of the advocate of applications of in this for the omics data a lot. So she write this review paper to summarize. If you have the sample which is normalized, okay, transformed the matrix, so it's ready to be decomposed. If you use NMF, you get genes and factors. Okay, so people use different names. So here we use factors, components. They all largely the same. Okay, so meta-gene, whatever you call. So you decompose this into gene factors matrix, and then factors by sample matrix, and then she made another name. This is called amplitude matrix. So just how much a fraction or how much this gene involved in each program or each factors. And she called this pattern matrix or the loading and whatever you call it. Again, it's just tell you each sample and how much this sample contribute or how much each of these factors contribute to the final expression total in that sample. Okay. So I hope you are completely familiar with this expression now. So you have small numbers of factors. Okay, in this case, the three factors. And when you do this multiplication between these two matrices, and you get the observation. All right? Then most recently, there are another applications which is inside using single cell data as a reference to decompose bulk RNA-seq data. Okay. Let's see whether this is. This is not the best expression of this problem, but it's okay. So maybe we use this. So there are a program called CyberSort has been here many years, so 10 years already. The idea is to use single cell expression profile we learned from individual single cell studies to build a profile of different cells. Okay, so if you have these 4 different cell type, and then you have single cell data from their expression, you can make a matrix. Okay, so this matrix contains information of different types of cells: macrophage, B cell, T cell, dendritic cell, and their gene expression of the genes. We can get this very easily, right? So now we will consider different cell type as factors. You see we add another terms here, cell type as a factor. So this have a lot of different definitions of what that factor is, depends on applications. So if we consider different cell type as factors, we can use this matrix to help to decompose bulk RNA-seq example observation matrix. So the idea is this is the observed. So this X is a bulk RNA-seq matrix which genes. So this X genes by samples. So we want to approximate this by a U and a V. So U is we build single cell gene RNA-seq single cell cell type gene expression profile matrix. So this will be, see, number of genes. That can be the same, okay? Same of set of genes. And in these columns, those are the cell type. So this is a U. So this problem is slightly different as you just want to decompose this, because if I not giving you this, you will decompose this into any two different matrices. But now I have some constraint. I know one of the matrix U it looks like this. What I want to learn is my V. Okay, so I want to get the values inside here. So what this V matrix should be. So what's the row? What are the columns this should be? So in order to make this inner dimension match, so here you have to be the same as cell type. So here's cell type, so you can make matrix multiplication. Well, in here, the columns are the samples. So you know all the numbers here, you know all the numbers here, so you want to fill up all the values inside this brown matrix. And that's what you want to learn. You want to know each sample, the values of this. Bigger indicate the sample with have a higher cell type in there, the fraction. Okay, so some samples maybe only have cell type 1, 100%. Sample 2, maybe cell type 1, cell type 2, 50-50 mixed. So you can see this heterozygosity or heterogeneity levels within each decomposed samples and their fraction of different cell types. Okay? So that's an application, right? Are we okay with this? Okay. So this make a link. So non-negative factorization help you to link single cell expression matrices with bulk RNA-seq matrix. So that's all. That's only the principle, the algorithm behind all this different kind of this, what they call deconvolution. Okay, we have a name. This procedure is called deconvolution. I'm not so sure whether it's O A. The deconvolution. It should be A, right? No, it should be O. O, yeah. So if you search literature and the deconvolution of the bulk RNA-seq, you are actually talking about this procedure here. Okay. So CyberSort is one of the earliest implementation for deconvolution algorithm. So people use it. There are several others which have a similar design. And this is one of the deconvolution algorithms. Then this matrix is marker genes. Okay, this is alpha, beta, gamma. gamma delta cells, and those are the expressions. So this matrix here on the screen is this. Okay, so this can be used if you have samples collected from bulk RNA-seq from pancreatic. Okay, you cannot just use random, nothing related. It has to be same tissue, and you know your bulk RNA-seq is a mixture of those individual cells. Yeah, question? If you don't believe the cell composition changed, there's a new type of cells comes in. If you don't have those major change, you should be able to use just the same reference. Right, just to see if yeah, the components change the fractions in each sample. Yeah, it should be. So this is an example of another similar, just different names, and the claim it may be more generalized, so you don't have to pre-select the cell type, and then it can also deal with this cross-subject heterogeneity. Yeah, but same idea at the end, okay? So just some different tweak of the algorithm, different intermediate steps to make this stronger in some aspects, but that's all. And more recently, there are applications of this framework not only deal with bulk RNA-seq, but also the spatial transcriptome. The reason we need that because the resolution of those spots looks very small, but under each of this probe, each needle, okay, it's a 3D tissue, so you probably spot many different cells at one spot. One is the resolution, another is a sample is 3D, so each sample is always a mixed. You don't have a single cell level resolution. So in this case, we need decomposition, okay? We need this deconvolution algorithm with the reference to tell you under each of the spots how much is fibroblasts, how much is immunocells, or is fibroblasts combined with cancer cells. So you need to have this deconvolution algorithm to tell you the composition of each spot. So this is one of the applications people nowadays use NMF a lot. Okay, any other questions? Good. So this is an illustration for this cell to location algorithm. You see this matrix is this, right? Cell type and gene. It's a U matrix. Then you have a different location. You believe each spot is a mix of different cells. Okay, in this case, 2 of this from one cell type, and this is slightly pure. So you get the estimation of what cell type in that spot. And then after you finish, you can see, okay, I want to look at the cell type 1. So this cell type, the fraction of this cell type is higher in this corner and slightly reduced in the middle and then dropped to this cell type. Well, in the middle region of this distribution, this slide, there are spatial information about those spots. So you can reconstruct this picture of what the cell type distributed in which location across this particular piece of tissue. Another package called the Spice Mixed, and they also use this image files, okay, fish files. You can have the information from different genes. You can build expression profile, gene ID, to do the decomposition. It's called metagenes, okay. Same idea, okay, looks fancy, but eventually are the same. So NMF is in the middle of here. Okay, so we will move to the next eigen decomposition. So we have talked about this eigenvalue, eigenvector, okay? Remember, this has to be squared matrix. You can decompose, you can have this relationship, you can find eigenvalue and eigenvectors to express the relationship like this. So this is an example, okay? You have the A matrix and its eigenvector V. So the multiplication between these two becomes this, okay? So that's the left side of the equation. This is this part. Well, this can also be expressed as the eigenvalue 6, which is lambda times eigenvector. So this is the right side of your matrix, and you get the same vector to satisfy this relationship. Okay, so we know this is a definition of eigenvalue, eigenvector. And we also know the geometric interpretation. So you have to think about A as a rotation matrix, and V is a special data point, a special vector in the space. And you use A as a rotation matrix to rotate V. V will not change the direction, but only the magnitude, how long the norm of V is, by a factor of eigenvalue. So now we have this equation, we will rearrange it to write, so this is the interpretation. We rearrange this relationship by putting eigenvectors from this end to this side, okay? So when you put this to here, okay, this is only one vector. Normally we have multiple eigenvectors. So in this equation we write in the matrix form. So several eigenvectors combined into a vector set. So make a matrix form. So once you have a matrix form, and this is a list of eigenvalues. So if you have multiple eigenvectors, you have corresponding multiple eigenvalues. So you have to write these eigenvalues into a matrix form, which just put them into diagonal position, make it become vector. Well, you remove this from this side to here, and you have to take this inverse. Okay, so just similar as a mathematical operation, just change the expression. And then you get this general formula to decompose A. Okay, you have A and you make them. You have X. You make them become a product of these three matrices. Follow the definition of eigenvalue decomposition. The eigenvalue, eigenvector. So you can replace those notations and write to replace this, and write into this way. Okay. So this emphasizes the if everything is real numbers here inside A. So in real application we have always a lot of this real number symmetric matrix. If that's the case, then the inverse and the transpose are the same. So you can replace the inverse with the transposition. Okay, inverse is much harder to compute, but the transposition of matrix is you don't need to compute anything. So if you have A is symmetric and also the real number, so this decomposition becomes much easier, and because you don't have to compute the inverse of the eigenvector. So that's the general relationship. If you don't have the symmetric, you have to compute the inverse rather than Q and the transpose. Okay. Diagonal matrix. So they're all just definitions and expressions, and we will play with in MATLAB. You can get idea about all this. Okay. So so eigen decomposition can help you to do a lot of things. First, for front line of the application will be just inside linear algebra. For example, can help you to compute the exponents of the matrix much faster. Okay. So if you have a matrix A, if you have compute the squared, you know it's A matrix multiplied by A itself. You know how to do the matrix multiplication. Basically, you have to compute a lot of multiplications and submit for each element. So this is relatively easy, but if you have a power to 3, 4, or higher power, then the computation becomes much expensive. In that case, you want to first to decompose A into, using eigendecomposition, into PD P inverse. You only compute once. So once you have this, you know you can, once you do this squared, you can just multiply this 3 by itself. Turn out this can be simplified because those cancel out. Okay, inverse and the matrix itself canceled out. At the end, you can express everything just using this D matrix. D matrix is very simple. D matrix contains only values in the diagonal position. Those are the eigenvectors, eigenvalues. So to compute that squared, it's just a series of numbers squared. So it's highly efficient. And then once you finish this, you can multiply it by this 2 eigenvector matrix. Then you get the results. So this is the same. But when you have a higher power, so for example, 25, for example, this is hard to compute. But with this, you just compute 25 power for D, and what's just contains single values, individual values. It's very easy. So this decomposition allows you to compute the power of matrices in a much faster way. Okay, so that's the power matrix exponents. Another applications in. Okay, so let's play a video. We are moving to the single value decomposition, SVD. Let's play this and then take a break, come back to discuss more about that. Okay? So this is application, highlighted application of the SVD in the engineering computing.
To a mathematician,
let me make this bigger.
Get up to 60% off thousands of items. Shop Michael's lowest prices of the season today.
Oh, spice. Super Hydration Body Wash. The sweet scent of a honeymoon in Fiji. Moist.
to a mathematician, a matrix is a table of numbers which describes the relationships between the components of various kinds of systems. The system might be a bridge, an electrical circuit, a nuclear power reactor, or a sociological group. Each of the components of the system, the bridge struts, the resistors and capacitors, the neutrons, or the individual people, interact with the other components. The matrix elements represent the intensities of all these interactions. Analysis of the system using matrix techniques allows one to predict the effect of individual interactions on the overall system. Oscillations may be generated. Matrix analysis can describe the frequency and magnitude of such oscillations. This bridge near Tacoma, Washington, was designed and built before the effect of such interactions could be completely analyzed. The wind has reinforced some undesirable oscillations with unfortunate results. Modern computers are particularly well suited to solving problems involving large matrices. The designer of a system can obtain accurate predictions about the reaction of his system to external forces. The computer is used to simplify the matrix so that most of the entries will be zeros. One method is known as the singular value decomposition. The original matrix is represented as the product of three matrices. In the second of these matrices, the only non-zero elements lie on the diagonal. These elements, the singular values of the matrix, determine the frequencies of oscillation of the original system. The other two matrices must preserve the properties of the original matrix, so they are orthogonal matrices. Each is the product of elementary matrices, which affect only two rows or columns. Scientists at Los Alamos Scientific Laboratory have been studying methods for the accurate and efficient calculation of the singular value decomposition and of describing to computer users how the computation is carried out. In this three-dimensional representation of a small matrix, each pyramid depicts the magnitude of a single matrix element. The diagonal of the matrix runs from the upper left to the lower right. Orthogonal transformations will be used to introduce zeros into the matrix. The initial transformations put zeros into the first row and then the first column. Each transformation affects two other columns or rows shown here in blue. Later transformations introduce zeros into later rows and columns, while preserving the zeros already obtained. It is not possible to annihilate all the off-diagonal elements in a column, so this first stage produces a matrix with non-zero elements directly below the diagonal, as well as those on the diagonal. This film is produced by exposing a single frame each time a matrix element is altered, to illustrate roughly the computer time actually required for various phases of the computation. The elements shown in red are involved in auxiliary calculations which require time, but which do not directly alter any elements. As more zeros are introduced, the rows and columns become shorter, and each transformation requires less time. The second stage of the calculation is a repetitive or iterative process which reduces the size of the remaining off-diagonal elements, but does not zero them in a single step. During the process, new elements, shown in red, are temporarily introduced into the matrix. The calculations now being shown are primarily intended to reduce the size of the off-diagonal element at the lower right-hand corner. Once an off-diagonal element has reached a negligible magnitude, it is set to 0. The computation of the corresponding diagonal element is then complete, and the resulting singular value is shown in blue. During these iterations, most of the other off-diagonal elements are also being reduced in size, although not as rapidly. A few of them will occasionally increase in size. This makes theoretical analysis of the method difficult, but does not seriously impair its overall effectiveness. Since elements are being depicted according to logarithmic scale, the off-diagonal elements we see are actually quite small by this time. The transformations are still changing the diagonal elements, but the changes are not visible on this scale. Once a few of the singular values have been obtained, the off-diagonal elements have become so small that each of the remaining singular values is obtained with a single iteration. The final diagonal matrix, together with the two transforming matrices which produced it, then provide the basis for a complete analysis of the original matrix and the system which it models.
You know Harbor Freight for a
Okay, so SVD. So if you are in engineering, when you pass by and look at SVD, you will stop and stop on it. But in the vet school, nobody cares. They just walk by. So this has been, the method has been discovered over 100 years. So there's a review paper. This discuss the discovery. And the video you just watched is a development of SVD, the practical computing method to compute SVD in 1960s. Then this algorithm is in the center of a lot of these linear algebra operations. So it's inside this linear algebra package, LAMPAC, and incorporated into MATLAB and other languages. So this is a widely used most known, okay, widely used. So this lecture a little bit upside down. So normally people will introduce SVD first, then non-negative factorization, because that one is much later, as I said. Okay, compared to eigen decomposition, this is a more stable, computational stable. Looking for my, yeah. And apply to any shape of the matrix. Remember, eigen decomposition only applicable for squared matrix. Well, SVD, you can have a matrix any shape, okay? To decompose this into three matrices. U, sigma V. And the people normally put a transposition here in order to, I don't know, but people just do that. So in the example, this A is a 2x3 matrix, okay? So if you have a matrix with n by m, and then this is a rotation matrix. So this one will be n by n. Well, you can consider this as a pairwise relationship matrix between rows. Well, this one is square matrix m by m. You can consider, let me use different colors. Make this better. So U here, n by n. You can consider that V matrix as a pairwise comparison of similarity matrix between variables. Okay, you can transpose whatever. What in the middle? So in order to make this matrix multiplication work, you have to make this matrix what's the shape of this. So this has to be n by m. Okay? So when you have n by n matrix multiplied by n by m, this becomes n by m matrix. Then inner dimension matched. So at the end, this becomes n by m matrix. So make this work, because original A matrix, let's see, A matrix. is n by m, right? Okay, so this is a rotation matrix and also normalized. This is also rotation matrix normalized. Normalized means the norm of each column is 1. So it represents a point in the unit sphere. It's a 2D, it's a point on the unit circle. Remember unit circle? We have tried a lot of times. See, okay? Any point on the unit circle has the norm equals to 1. Okay? So this point, which for example in the U matrix contains a vector of these two, I'll tell you the direction. So those norms. The vectors are on the unit circle. I believe those vectors also has to be orthogonal to each other. So in order to be inside the U matrix, these two vectors has to be 90-degree, make a set. Okay? So what's this set? Is x, y axis. Basically, this is a coordinate system. So this represents a coordinate system. If you have a 2x2 matrix, it will be a set of two vectors on the unit circle to define a coordinate system. If you have a high dimension, still the same principle applied. So U and V are two rotation matrices to help you to rotate your data, but not change the magnitude, not the shape, only change the angle of. So you know this in the PCA study. So in PCA, you want to get a rotation matrix U to rotate your data points. But this U matrix has to be normalized. So when you multiplicate your rotation matrix to the data, you don't change the magnitude of the data to the origin. You only change the relative angle of observation by changing the coordinate system. Rotate this. Another thing you have to pay attention is this sigma matrix. Okay, this sigma matrix is in the middle here. It only contains values in the off diagonal, in the diagonal position. So you have this 1, 1, 2, 2, and because this matrix is not a 3 by 3, so you don't have any values here. So you just fill up the last column with zeros. But the most important ones is only in this squared part of the matrix. So if you have n smaller than m, then the only part in this matrix will be n by n, which have values. The rest of them are just zero fill up to make the matrix to the dimension of m. So this is a construction of these three matrices in the SVD. So this is what I'm writing on the board here. Okay. And this is a representative example. This is a video explain the rotation matrix. This, that. Okay, this is the best video I can find in the YouTube. It explains that SVD decompose a matrix into 3 components, and these 3 components contains 2 rotation matrices, which expressed in here. So rotation matrix U, rotation matrix V. Okay, so that's A is original and has been decomposed into 3, and this 3 can be translated into geometric interpretation. See, okay, rotate your vector. Okay, in this case, this rotated vector rotated the points into here, so nothing changed. And then the scaling factor, the matrix, will change the magnitude. Okay, so this case make component 1 longer, component 2 shorter. And then this A contains another rotation matrix U, and you can rotate that again. So the transformation operations which encoded inside this observation matrix A contains 3 components: rotate, stretching, because by introduce the change of magnitude in different dimension. So stretch the shape of your data and rotate again. So this contains 3 steps. So if you do the opposite, if you do this first, that again, you will transform this back to the original. So I will play this for 3 minutes.
This week, we are changing topics to, I'm going to go out and say it, this is the most important week of the class. What you learn here is one of the most important tricks of the trade. It's called, I'm going to write it up on the board, the singular value decomposition. This week's lecture's focus on this is going to basically introduce you to this really amazing data analysis tool. It's all linear algebra, as always. But what you can do with this is quite remarkable. So in this first lecture, I'm going to introduce you to this singular value decomposition. And by the end of this week, I'm going to show you how you can use this to do things like start thinking about how you might do image recognition software for faces, for classification, dogs versus cats, and all kinds of interesting ideas that come out of this singular value decomposition. Extremely powerful, extremely useful in a lot of contexts. And so let's begin, and let's start thinking about what this thing is. So
yeah, so this is like a 1 week introduction, but these videos have been very popular, and you won't be able to miss it if you search singular vector value decomposition, and I suggest you spend some time to watch it. Okay, so examples. So in this example, we consider there's some books. Okay, so this book are the samples. We know they have several different books, and there are 2 major categories. So we can truncate, basically we truncate this not into the n by n, but use n by 2, okay? If you don't do any truncation, you will get, depends on how many, so here 1, 2, 3, 4, 5, 6, 7, 7 by 5. So you should have a 7 by 5 or 5 by 7. So you should have 5, 7 by 7, 5 by 5, something like that. This example is not the best for the illustration of this, but it's a truncated version. If you know this matrix contains only 2 components, the most important ones, you can approximate that by 5 by 2, and here will be 2 by 7. To decompose that only use the most important components, okay, to reconstruct this. If you have no idea of this number, you can do the full spectrum decomposition. To decompose it into 5, 5, 5, 7, 5, 7 in the middle, 7, 7. And then you will discover that a lot of these values which above 2 dimension are very small. It's close to 0. So you know they are dispensable. You just throw them away and only use the top 2 components to reconstruct the whole thing. So this is a truncated version, and to show you that you only use 2 components to reconstruct the whole matrix. So this is 7 different books, and with those words, okay? The computer science book contains, this article contains data and those frequencies. This data has not appeared in the medical related literature, while the brain is only appeared in medical related literature, not in the computer science. And then we learned the first matrix is sample by sample, then only talk about the first 2 samples, components. Then the sample, the first couple of samples have this component 1 strong, and the rest of the articles or books have the second component strong. So there's opposite patterns here. And inside this is the signal matrix contains the strength of this concept. So this concept is what we talk about. We have 2 concepts or components. Depends on what you call, depend on the application situations. So the terms 2 components matrix is the last. So we decompose that, the frequency of the words in the 2 sets of different types of the literatures into these components. Each component, this matrix has its own meaning of those, okay? So this is a decomposition of this A matrix into U, sigma, and V. You can compare this strengths of each components, see their relative contribution to construct this. You will see this is much larger, okay, compared to this 0.7. So what that means is you can use the only first components to reconstruct the matrix without losing a lot of information, because the second components only contribute 0.7 in terms of strength. Explain. What you can consider is A matrix has 2 layers. The first layer could be 0.8, 1.8, 2.3, something like that, which you only need to use the first components to reconstruct it. What you can do is to use this column, multiplicate by this value, and multiplicate by this row. Maybe this column, because it's transposed. So I can give it a try in MATLAB to see whether I can use the first components to reconstruct this very close. So I want to use the first component U, so U1 is negative 0.39, negative 0.92. Okay, this is my first. My sigma is the value 9.51. Well, the last V1 it's negative 0.42.81. You see the full decomposition, you need to have this full matrix to reconstruct it. But because I know there's major contribution is from first components, so I just extract the most important components and using the part of this information to reconstruct the original matrix. Let's see, I can use U1 times S, so this will give me 1, 2, and so I need to transpose one of this. V1, I think transpose this. Incorrect dimension. Let me do that again. So this is, I probably used the wrong dimension of the information, so it should be 5, 7. Yeah, so if you look at this reconstructed matrix, it's not too far from this original A. So the first is 1.5, there was 1, and the second is 2, this 2.1, and this third is 3, and my reconstructed is 2.59. Yeah, so what I want to compare is the matrix I reconstructed using just first components is almost reflect the original observation I want to reconstruct it. And then I should add another value and another set to make this exactly the same, close. But this just give you idea. I only use the first components to reconstruct the original, because I learned from this single value, singular values, the magnitude, the difference. Okay, so this tells you the truncated version. So you have a full version of this decomposition, but when you do the opposite, you only use part of the information. So you truncate the most important ones and use this simple ones to reconstruct the original. So this procedure, the reconstruct, this decompose and through truncated version and then reconstruct is called low rank approximation. We do this to do the denoise because it's in the original matrix. If you have some small noise, in order to reconstruct the whole thing accurately, you need to introduce a lot of the small contributing factors in order to get the full reconstruction. But if you know those are the least important ones, you can just throw them away by looking at the singular values, by the absolute values. And then you can throw them away, use the clean or major contributing factors to reconstruct this observation. The data become cleaner, so you throw away those low frequency noise. Same thing to illustrate, we use the truncated version and just major ones, 2 of them to reconstruct the original one, which have m and n dimension. This is different ways to write this. So this is use truncated matrix you get from the decomposition, but you can also just write each individual vectors from the reconstructed vector dimension matrix. So this is what I just used one vector, one dimension to reconstruct the 1, 2, 3, the matrix. Where you can write in this this way because this created the first. Let me use this. So this created the first layer of, if you consider this as a multi-layers of matrix put together, and this reconstruct the first major layer, the most important ones. Then this is second one. You may have a lot in order to reconstruct this, depends on how noise your data is. Then you can throw away those least important ones to get approximation, which is the noise version of the A. So how SVD is computed? Using the built-in function, you can have SVD function in MATLAB and give A matrix. A matrix can be any shape, and you always get SU, SV, the 3 matrices. The algorithm, as I said, is very stable, so we need to understand a little bit of what's inside this function, okay? That's a Julia version and the same thing. So this is a contribution in 1960s by this Professor Gene Golub, and he's a professor in UC Berkeley. And this is his plate of car, Professor SVD. His contribution has been built into this Fortran source code to do this as a foundation for the LAMPAC package, and MATLAB is using this, and many other high-performance computer language also. At the bottom, if you search, at the end is this. So how this was computed using several methods? I think they implemented using the QR divert. Some of this I can't remember exactly. So this is one way. There's a normal matrix to do this. Then there's another way to do this block matrix. So this is an indirect method. Is the most intuitively sound. I found it's very easy to understand. So what you can do is you transpose your matrix, okay, multiplied by itself, the transpose, and basically you get the covariance matrix if you consider this as variables. And then you do the same thing for you multiply it itself by its transposition. Basically, you get a Gram matrix. You know, the covariance matrix, Gram matrix, they are related, but one is for between sample to sample, another one is between variables. So you write in this way of if you decompose this into three matrices, and you can rewrite A into the product of the three, and you do this operation and you express this with a V matrix, okay, because this U has been cancelled out. Then this becomes, to solve the V, that becomes an eigen decomposition problem, okay? So V basically is an eigenvector once you do by M Gram matrix. And on the opposite side, this side, the U is eigenvector for between N by N matrix, correlation matrix. So in this way, we can compute both sides in terms of variable matrix, variable to variable matrix, and the sample by sample matrix become two eigen decomposition problem. So in this indirect method, you can solve this using two eigen decomposition methods. So SVD can help you to accelerate a lot of computing as well, and this is to do the so-called pseudo inversion, because you can just inverse the only inside of the S matrix. So to do the inversion of this is very easy, just 1 over the numbers of those, because this is the only have values in the diagonal position, and the UV is already computed from out of this SVD. So this makes the inversion computing very efficient. You only do SVD and then you inverse diagonal matrix. So this is just to show you in different ways we can compute this and reach the same results. And remember the inversion of this X matrix, we used it in the regression when we compute regression coefficients. Okay, the last 5 minutes will be introducing the independent components analysis. So this is set up in slightly different ways. This is to solve a problem called a cocktail party problem. Okay, so you went to a cocktail party and with all people with background music, and there are 2 microphones will be put in the different corners of the room, and one microphone will be close to the music, which far away from the people conversation, so voice, and another will be put in different corners, which close to the conversation, far away from music. So these 2 microphones sample the same set of the mixtured sound, but in the different proportion of different source. So if you have this data and collected by independent, these 2 source microphone, and then you can use ICA to separate the background sound which generated from the music and also conversation to basically decompose or separate the source, independent source from the mixed information. So you need 2 microphones. If you have only one microphone collect the mixed sound, you don't know the 50-50 how you separate them. But if you have 2, you should be able to separated the two major sources. But if you have 3 sources, you may need 3 microphones to collect different mixture, different components of this. So this algorithm try to rebuild a mixture matrix. So this mixture matrix A will be approximated by the W. So it knows, okay, microphone 1 is collected the conversation 90%, 10% from music. Microphone 2 will collect 20% of conversation and 80% of music. So it will reconstruct this mixing matrix to help you to understand the independent source. And then a lot of algorithm has been developed, and we can use this to do a decomposition of the mixer information. So this is a collector of this EEG signals from multiple probes. Okay, they put the probes in the head at different regions, and then to learn the major sources. By using this algorithm, you can separate them. We will play examples in the computer session. We have a mixer sound, and after you put that mixer into the algorithm, we'll clearly separate the background noise on the street and with also the different source of the conversation. All right, so this is the last decomposition method I will introduce for now. All right, see you Thursday to do the computer exercise. Have a good one.